Hello, my name is Nicholas Reyes, and this local expert project is on stem cell culturing. First, going over the technique explanation. To begin, this technique is done with stem cells. What are stem cells? Stem cells are cells that have the potential to develop into many different types of cells in the body. The objective of stem cell culturing is to differentiate the stem cells into the cells that are necessary for the project you are working on. Stem cell differentiation is triggered in vitro stem cell cultures that can replicate the environment in vivo. This technique uses different media and substrates to trigger these differentiations. A carefully crafted and controlled environment to support the growth and direction of the cells are to be grown into are needed for this technique to work correctly. Stem cell culturing pertains to Module 5.1, as 5.1 is the module for cell culturing. This technique is a technique that is used for stem cell research, which is a growing field and is necessary to work on research such as cancer or brain illnesses. Now, what are the limitations to stem cell culturing? First off, durability. Adult stem cells have been shown to not be as robust and durable as embryonic stem cells in comparison, which can be seen as the alternative to stem cell culturing, as these are different types of stem cells you can culture. The second is limited variability. Adult stem cells may not always be differentiated into all types of cells. This can lead to limitations to what can be studied and what can be used to treat diseases. And lastly, more susceptible to abnormalities. Adult stem cells are more likely to contain abnormalities due to the toxins or errors in the replication process. What are the advantages to stem cell culturing? First off is flexibility. Since you are working with cells that can differentiate into other types of cells in comparison to other culturing techniques, you have the range to differentiate into various cell types and not just the one you cultured at the very beginning. Reduced ethical concerns. Induced pluripotent stem cells, or iPSCs, offer a method in which can be used alternatively to embryonic stem cells, which are other types of cells you can culture that may have some ethical concerns attached with it. Patient-specific cells is another advantage to stem cell culturing. As with the reduced ethical concerns, iPSCs can be derived from the patient's own cells to allow for a more personal therapeutic approach if necessary. Now, what are the controls you would use for stem cell culturing? The controls that would matter in stem cell culturing would ultimately depend on the type of cell you are trying to differentiate into. Altering the environment of the stem cell culture would change what is grown and what it needs as well to grow. Changes such as environmental cues, such as growth factors and cytokines, and specific signals and cell-to-cell interactions would need to be taken into consideration as to what controls are being used for your stem cell culturing. Now onto the three figures from the three separate articles that use stem cell culturing. The first figure shown on the right. In this figure, it is showing the characterization of human iPSCs, or induced pluripotent stem cells. Part A shows a micrograph of parental fibroblasts. B shows derived iPS. C shows control human ES colonies. And in the graphs below, in H, it is showing the gene expression patterns of the cells in A through C. Now for figure 2, on the right-hand side, what this figure shows here are multi-scale biomaterials to control the stem cell behavior. Now there is a lot going on in A through E, but taking apart and looking at specifically part D in the figure, for example, in figure 2D, a method to control stem cell behavior at a macro scale by 3D printing organ shape and tissue function, such as embryonic hearts, can induce specific differentiation of the stem cells you are trying to culture. Last but not least, in figure 3, this shows in 3 parts the differentiation potential of mesenchymal stem cells, or MSC. In figure 3a, this is after staining for lipid vacuoles, and 3b showcases the formation of mineralized matrices. And in 3c, this shows that these were positive for chondrocyte matrix. And with these three passages, this shows that there is adipogenic, osteogenic, and chondrogenic differentiation, respectively. Thank you, and I hope this presentation shed light more on stem cell culturing.